In this video, we'll be diving into how gene function is explored through knockout organisms, how CRISPR is revolutionizing gene editing, and why some DNA sequences have barely changed over millions of years. Sometimes the best way to understand a gene's function is to delete it. Gene knockout is a method used to make a gene inoperative, like switching off a light to see what goes dark. Scientists create knockout organisms, usually mice, yeast, or zebrafish, by removing or disabling specific genes in their genome. Then, they observe what changes in the organism's phenotype. If a knockout mouse develops diabetes, it suggests that the removed gene plays a role in insulin regulation. Entire libraries of knockout organisms exist for research. For example, there's a mouse library where each strain has a different gene disabled. These are powerful tools for uncovering gene functions, modeling human diseases, and identifying potential targets for treatment. CRISPR-Cas9 is one tool used for these genetic modifications. CRISPR-Cas9 is a tool that makes gene editing faster, cheaper, and more precise than ever before. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. It originally comes from bacteria, where it functions as a natural defense system against viruses. CRISPR and Cas proteins allow bacteria to remember viruses they've encountered in the past and essentially destroy the virus by cutting the viral DNA. It is used in a similar way by scientists to cut and modify genes in other living organisms. CRISPR sequences are short, repetitive DNA regions. Scientists pair them with an enzyme called Cas9, which acts like molecular scissors. Together, CRISPR-Cas9 can be programmed to find a specific DNA sequence and cut it, allowing scientists to delete, add, or replace genes in living organisms. In 2020, researchers used CRISPR to treat patients with sickle cell disease, editing their blood-forming stem cells to stop producing the faulty hemoglobin. Early results were incredibly promising as some patients experienced less blocked blood vessel pain and improved hemoglobin levels. Scientists are still pushing the boundaries of using CRISPR-Cas9 today, making the process more precise and safe for therapeutic use. Of course, powerful tools bring powerful responsibilities. Some uses of CRISPR, like editing embryos or altering human germlines, raise major ethical concerns. Could it lead to designer babies? What are the long-term effects? And who decides what's acceptable? Different countries have different laws. That's why there's an international effort to harmonize regulations around genome editing. Scientists and policymakers are working together to ensure CRISPR is used safely and ethically. As IB students, it's important not just to understand the science, but to think critically about its implications. We know mutations lead to genetic variation, and the rate at which mutations occur is fairly constant and predictable. However, there are some genes which show relatively few mutations over time. These are called conserved or highly conserved sequences. Highly conserved proteins tend to fall into a few key functional categories because they are essential for life. These include metabolic proteins, fundamental reactions like aerobic respiration, structural proteins which maintain cell shape, genetic information processing proteins involved in DNA packaging, replication, or repair, or ribosomal and translational proteins which form the core of the ribosome and ensure proper synthesis of proteins. For example, the protein cytochrome C and its corresponding genes are highly conserved, meaning that cytochrome C has changed little over evolutionary time. Cytochrome C is a small, water-soluble protein that plays a crucial role in both cellular respiration and programmed cell death, or apoptosis. It is about 100 amino acids long. Pigs, cows, and sheep have identical cytochrome C molecules, amino acid for amino acid. Similarly, the cytochrome C of chickens and turkeys is identical, and ducks only differ from chickens and turkeys by one amino acid. In fact, researchers have inserted cytochrome C genes from fish, horses, humans, and birds into the genomes of single-celled yeasts, and the yeast have successfully produced the cytochrome C protein. There are two key hypotheses to explain this. The first is the functional constraint hypothesis. It states that the sequence is critical for survival. Mutations in this gene would disrupt the function of vital proteins or RNAs, so these mutations are eliminated by natural selection. The second hypothesis states that there are lower mutation rates in some regions of the genome due to being more structurally stable or more efficiently repaired during DNA replication. Both hypotheses attempt to explain why some genes have remained nearly unchanged across vast evolutionary timescales and diverse species.